I'd like to thank the organisers very much for inviting me to speak today. And so as Nicolai said, I'm going to give a little bit of a background on what we're doing here in trying to develop vaccines against complex pathogens. So as everyone in the audience knows, infectious and parasitic disease are a major cause of morbidity and mortality throughout the world. In particular, children under the age of 5 are at a major risk of disease from a number of these infectious and parasitic diseases. And it's really well established that the vaccines are a very effective control for infectious and parasitic diseases. They're one of the most efficient and cost-effective interventions for the control and eradication of diseases and the prevention of morbidity and mortality worldwide. And in fact, it's been demonstrated by the WHO through other efforts that no other modality other than vaccination has had such an effect on reducing the morbidity and mortality and improving public health, except for water sanitation. And also, vaccinology is actually the only science that has eliminated infectious diseases, which is a case of smallpox in 1980. So this slide here shows a pipeline that was put together by the World Health Organization a number of years ago. And what it does, it illustrates the traditional vaccines that have been administered with the EPI schedule, other vaccines that are already available which are potentially underutilised in a number of areas, and then the future vaccines for which vaccine development is urgently required, and in particular highlighting what they call the big three, the HIV, tuberculosis and malaria. So the other point that I want to make is that currently there is no licensed anti-parasitic disease for humans, despite the fact that infectious diseases are a major cause of mortality and mortality throughout the world, and also have a huge socio-economic effect. And also that there is no vaccine against any chronic disease. And there's a number of diseases which obviously fall into this category, including the parasitic diseases which we're looking at, but also viral and bacterial diseases. And there's no vaccine against any chronic disease caused by a complex pathogen. This is largely because such pathogens have adapted to long-term coexistence with their human hosts through, with the human immune system through coevolution over many, many, many years. They have, a consequence of this, they have developed sophisticated immune evasion strategies. And many of these organisms occur within the human host within multiple stages. They have distinct phases of their life cycle within different host cells. And many of them are very large and complex and can express hundreds or even thousands of potential antigenic targets. So malaria is a prototypical example of a complex pathogen for which we urgently need to develop a vaccine. As everyone would be aware, it is a major cause of mortality and mortality throughout the world, and as well as a huge socio-economic burden. It impacts not only people that are living in endemic areas, but it also impacts travellers and the military. There's currently no licensed vaccine, and there's increasing development of drug resistance in the parasite and insecticide resistance in the vector, further emphasising the need for the development of an effective malaria vaccine. So people have been trying to develop a malaria vaccine for about half a decade, and there's currently no licensed vaccine. However, we do believe that the development of a malaria vaccine is feasible because we know we essentially have 2 human models showing that this is possible. We know that mice, monkeys or humans can be immunised with the bite of Plasmodium sporozoites that are attenuated by radiation so that they can infect the liver stage but then not develop further. In this case, the immunity is directed against the liver, and we believe that the mechanism is T-cell mediated responses against antigens expressed in the liver stage. The second model is that of naturally acquired immunity, and we know that in areas where malaria is endemic, there is a decrease in incidence, prevalence and density of malaria with age. And in this case, we believe that the immunity is primarily mediated by the blood stage of the parasite life cycle, in particular with antibodies and probably CD4 T cells directed against antigens expressed in the blood stage. So we have these 2 human models, but nonetheless we have not managed to develop an effective malaria vaccine. And that's because the parasite is a large multi-stage life cycle, expresses about over 5,000 proteins, many of which could be a target for effective immune responses. Many of these proteins exhibit antigenic and allelic variation, And most of the vaccines that have currently been tested to date in preclinical development have been based on a very small number of target antigens. In particular, there's 4 that have been the major cause or major foundation of most vaccines. And we really don't know very much about the mechanisms of protective immunity against malaria, which is the T-cell and antibody-mediated mechanisms. And we have not yet been able to develop any algorithms which are able to predict from these lists of about 5,000 proteins which ones are the key ones that we need to target for the development of an effective malaria vaccine. So this slide is a short slide which gives an example of the global project activity for malaria subunit vaccines. And what it shows really is that there's been a lot of effort in the preclinical development and the early stage, but there has only been one single vaccine which has actually made it into the phase 3 trials. And this has been the RTSS vaccine, and there was a press release on Friday showing that in fact this vaccine, which has been developed by GlaxoSmithKline, has received positive opinion from the European regulators. However, this vaccine, to get to this stage, it's taken more than 40 studies, preclinical studies. There's been an enormous cost associated with the development of it, but in particular, this vaccine, although it does prevent the death in some individuals, what they call protection number of disease deaths averted, it actually is a very limited efficacy. So we really need to do something better than working on RTSS. So this failure of subunit, the traditional approaches to develop a subunit for malaria vaccine, has sent a number of the researchers back to the whole parasite approaches. So the Scenario and Steve Hoffman's company is trying to develop a vaccine based on the irradiated sporozoite model. Other researchers in Europe and Australia are working on the development of a genetically attenuated parasite vaccine where genes within the liver stage or blood stage are knocked out of the parasite. And other researchers, particularly Michael Good, also in Australia, is developing chemically attenuated parasite vaccines. But these whole organism parasite vaccines can almost be reviewed as going sort of back towards the traditional vaccine technologies which were really pioneered by Edward Jenner in 1796, where he immunised against smallpox by vaccination with cowpox. So really, in the last 200 years, going back to this whole parasite approach means that we really haven't made a lot of progress. However, the sequencing of the Anopheles haemophilus genome in 1995 started the era of genomics. And so now we have a large wealth of data from the genomes, proteomes and transcriptomes of many pathogens that are a major cause of public health issues throughout the world. And so essentially this genomics, proteomics and transcriptomics information can be considered to provide a foundation for a new approach to vaccine development. And in the case of Plasmodium, the genome, proteome and transcriptome were elucidated in about 2002. And what this showed is all of the malaria parasites are very large, as I mentioned, expressing over 5,000 genomes. So this does provide a foundation for a new approach to vaccine development, but the challenge is how to use this information to develop an effective vaccine. And this is where the field of immunomics comes in. So immunomics is a field which bridges the disciplines of genomics and proteomics by using the host immune system. It really tries to elucidate the set of antigens or epitopes that are interacting with the host immune system and the mechanisms responsible for these interactions. So I just want to point out that it is actually, immunomics is distinct from reverse vaccinology, which aims to develop the complete repertoire of antigens that an organism could display on this surface, because immunomics uses the immune system. So what we're using biologically relevant criteria such as immune reactivity and using specimens from humans that are either experimentally immunised or naturally exposed to the pathogen of interest to try and identify key targets that should be incorporated into an effective vaccine. And what we're doing is we're using two approaches. One is we're using sera and plasma from exposed or immunised humans to try and identify the targets of antibody responses. And then we're also using peripheral blood mononuclear cells from these individuals to try and identify the targets of T-cell responses. So really, immunomics is vaccine antigen discovery using biologically relevant selection criteria. And in my laboratory, we've been using two approaches. One, an antibody-based screen to try and identify the antibody targets, and also a T-cell-based screen to try and identify the T-cell targets. So in terms of the antibody screening, we're using protein microarrays. So this is a technology which is developed with Phil Feldner at the University of California, Irvine. And really what we do is we take a number of genes from the genome sequence of the pathogen of interest, we clone them into an expression vector, and then we express these proteins in a high throughput manner, which can take up to 2 or 3 days, and then it's a cell-free in vitro transcription translation system. And the proteins that are expressed in this system can then be printed directly onto a microarray chip, which is essentially the size of a microscope slide, and then these chips can be assessed for quality control using standard QC measures, and then can be probed with serum or plasma from humans naturally exposed or experimentally affected with the pathogen of interest. And then essentially what we do is we get a picture of immune reactivity, which can be analysed as similar to DNA microarray analysis. So this is just showing an example of sort of a protein microarray screening, and we've looked at samples from individuals through a number of geographical areas throughout the world. And what we're doing in here, I'm presenting data in this case as a heat map, where I've got, in this case, the array contained over 2,300 proteins. So each of these rows here is an individual protein. Each of the columns is an individual subject, in this case naturally exposed in Papua New Guinea. And what you can see here, looking at the immune reactivity, where green is no reactivity and red is high immune reactivity, we get a number of antigens which were highly recognised in individuals naturally exposed to malaria in this case, and not recognised in individuals that are malaria naive. And so we can then use this to identify many new immunoreactive proteins. So what I've done here is I've just blown up the top 100 immunoreactive proteins from some of this data, and this is individuals naturally exposed to malaria in Papua New Guinea. And what we can see is there are a number of these individuals that are highly reactive. And what I've pointed out here is the antigens which actually are present or have been tested as potential candidate vaccines for malaria. You can see here that we've got many of these are really novel antigens, and CSP, for instance, and SSP2 or TRAP, which are other antigens, are ranking way down on the list here. So really what we're finding with the antibody screening, about 30% of the proteins are seroreactive, and we've identified a number of novel antigens which are more reactive than conventional historical antigens. We're also able to analyse these data with a number of different bioinformatical and biostatistical approaches and identify different features. And one of the things that we've been able to do, for instance, is to identify a panel of 16 antigens which appeared to be associated with sterile immunity against malaria induced by immunisation with radiation-attenuated sporozoite. And what we found in this case is that the antigens that were within this panel of 16 antigens, the serodominance does not correlate with protection. So in other words, the most reactive did not come back as being associated with protection. And we also found that it is an accumulation of immune responses to all 16 of these antigens, not to any single antigen, which does correlate with protection. We've also applied these protein microarray screenings to samples from individuals in endemic areas. I'm showing here is data that was conducted in association with Peter Crompton and colleagues at NIAID. And in this individuals in Mali, what we were able to do was identify 46 signature antigens which appeared to be preferentially associated with protection against clinical disease in the field situation. So what I want to do is point out here just the difference between malaria susceptible and malaria immune. In particular, highlight the fact that these antigens up here, which are the antigens which have been used in candidate malaria vaccines so far, have not really been able to discriminate between susceptible and immune reactivity. Whereas if you look at some of these novel antigens identified by protein microarrays, they are able to significantly discriminate between susceptibility and immunity. So these potential antigens look like they're good targets for malaria vaccine development. So just switching now to the T-cell based studies. And what we've done in this is a study that was funded by the National Institutes of Health. And in this case, what we did, working with Alex Sete and his colleagues at La Jolla Institute of Allergy and Immunology, we scanned the entire Plasmodium falciparum proteome to identify all of the proteins that were associated with the liver stage. So all of the proteins that we thought were expressed in the liver stage of the disease that therefore would be good targets of cell-mediated immune response. So we identified about 1,500 proteins. So for each of these 1,500 proteins, Alex's group then scanned them using computerised algorithms to identify peptide epitopes which were capable of binding with high affinity to distinct class 1 or class 2 alleles. So we identified 3 class 1 alleles and 1 class 2 allele, which together seem to cover all of the ethnic variation throughout the world. And for each of these HLA class 1 or class 2 alleles, we identified 5 peptides. And we synthesised these peptides, so we had a total of 29,000 peptides from this project. And for each of the antigens, so the 5 peptides for each of the 4 alleles, so a total of 20 synthetic peptides per protein, was screened for immune reactivity by using PBMCs from individuals, in this case naturally exposed to malaria in Papua New Guinea. And we could use this data to prioritise the antigens for immune validation. So I'm presenting the data here in a similar manner to the data I presented for the antibody screening. So it's a heat map of reactivity. For each of the antigens, we tested 10 individuals. So we've got the responders going down the y-axis here, and each of these across here is one of the individual antigens tested. And so again, what you can see is that there are a large number of antigens which are not recognised or which are very poorly recognised, and that there's a very small number of antigens shown up here which are highly reactive. And in fact, there's 12 proteins only out of the 1,500 that we've screened which were recognised by all 10 of the subjects. And most of the proteins, there was about a number of them were not recognised. The other thing I want to point out is, again, I've shown on this slide the CSP, which is the basic component of the RTSS vaccine, as well as other antigens which have been evaluated in preclinical studies. And so the take-home message here again is that you've got many, many more antigens which are completely novel, which look like they're really good targets. And this is shown here when I've just used as a cut-off all of the antigens that were recognised by more than 80% of the individuals. There were 80 antigens recognised by more than 80% of the individuals tested, and all of them novel antigens. And in particular, these ones up here, which are high recognition, high frequency and magnitude response, appear to be excellent, potentially excellent vaccine targets. So we looked at the antigens that we came up as highly reactive in our immune screening and tried to figure out what was associated. I just want to point out that the immune reactivity was not associated with this stage of expression. So Carla Proetti, who was a postdoc in my lab, was then trying to look at the data, look at any associations between the antigens which were identified by antibody screening or T-cell screening. And what we found out is that obviously the patterns of response is extremely complex. So this is just a circus map showing the frequency and magnitude of responses against both T cells in red and antibody responses in blue, which is extremely complex. So we've used a number of different methods to mine this data, and we've come up with some really interesting findings, such as the fact that there is no correlation between T cell and antibody responses. And in fact, there appears to be an inverse correlation. So in other words, if you look at some of the antigens which are not recognised at all by antibody responses, they're highly reactive for T-cell responses. Conversely, ones that are highly reactive for antibody responses are not recognised by T-cell responses. And in fact, there is a significant inverse correlation. So the antigens that are targeted by T-cell responses are not the same as those targeted by antibody responses. Another thing that we found was interesting, that we found that although the antibody targets were highly polymorphic, in our screening studies, the antigens that were targeted for T-cell responses appeared to be conserved. So we're just showing here we've divided all of the data into quartiles of high, medium, low or negative immune reactivity. And you can see that there is, this is in quartiles and this is showing the single nucleotide polymorphisms of the top 100 antigens. And you can see that the antibody targets are highly polymorphic, whereas the T-cell targets are conserved. And again, indicating that these antigens, which are identified by our immunomics T-cell strategy, are conserved and potentially therefore excellent targets for vaccine development. So we also looked at the data in many other methods, including a principal component and components analysis and a redundancy analysis. And what we were able to do was to show both that the antigens that were recognised by both T cells, shown in blue, and antibodies, shown in red, appeared to be very distinct. As you can see here, using the redundancy analysis, that there's no overlap between the T-cell targets and the antibody targets, and also by the principal components analysis. So this suggested to us that there may be biochemical and structural features that would allow us to identify from genome sequence information, proteome information, the key antigens that would be associated with antibody or T-cell reactivity. And so what we did was we identified a total of 69 different features, genomic, proteomic, population biology, many other features. And then we screened the antigens for these features. And what we were able to do was to identify a number of features which were highly correlated with antibody responses or highly correlated with T cell responses, or inversely correlated with antibody or T cell functions. So in other words, using computerised algorithms and bioinformatical screening, we were able to identify a number of structural and functional ideas which appeared to be able to predict antibody and T cell reactivity, and so therefore could potentially be applied to many other pathogens, not just malaria, but many other infectious disease pathogens. So just to summarise, I hope I've been able to show you that the immunomic approaches and proteome-wide data sets of antibody and T cell reactivity, we believe, do allow for identification of novel antigens which are targeted by protective immunity. In the case of malaria, we've shown that only about 30% of the proteome is highly recognised for antibody responses or for T cell responses, and in particular, a large number of the antigens are not recognised. We've also shown that not all antigens are equal, that there really is a hierarchy of antigenicity, immunogenicity and protective efficacy for both antibody and T cell responses. We've been able to identify many novel antigens that which appear to be far more antigenic than historical vaccine candidates. And significantly, we've shown that there appear to be distinct sets of antigens which are preferentially targeted by either T cells or antibody responses, which really means that if you're trying to develop a vaccine based on T cell responses, you want to make sure you use antigens that are recognised by the T cell responses. Trying to use antigens that are serologically dominant will not be the most effective use of vaccines. And we've also shown that the antigens which are targeted by antibody responses are polymorphic, but that there is at least a subset of really highly antigenic antigens for T cell responses which are conserved not only between species, strains, but also between species. And finally, we've been able to identify a number of molecular and biochemical features which appear to be able to discriminate between antibody and T cell targets, and that can be potentially applied to many other pathogens. And I haven't shown the data, but some of the antigens that we have identified by these screening methods we've shown in animal models are very good targets of protective immunity. So just finally, I just want to thank the many people involved in this work, in particular Carla, who did the antibody T cell studies, Angela, who did the protein microarrays, Joe and Bruno, who did the T cell screening, and Lutz for doing the algorithm predictions, Phil Feldman and his colleagues for collaborations with protein microarray, and Alex and his colleagues for collaboration with the T cell screening, and the many individuals associated with providing samples for these studies. Thank you.
So the paper is open for discussion.
It's my understanding that even after infection or many infections, we will only get CMA immune towards malaria.
Sir.
You will never get complete immunity or protection against malaria disease, even after infection with malaria. So my question is, why do you think that the vaccine could protect you against malaria?
So the immunisation with radiation attenuated sporozoites has been shown in mice, monkeys and humans to give sterile immunity. So if you inject the sporozoites that are attenuated by radiation, you can get sterile protection. And that's what the Scenario vaccine, the Scenario company is trying to develop. So we believe in the case of the T cells, you can get sterile immunity. But the other side of the developing a malaria vaccine is that people have been trying to develop a vaccine based on the dominant antigens which are exposed to the immune response, and it's likely, particularly in the case of T cell responses, that it's antigens that are not highly dominant for the immune reactivity, which may be really good targets for a T cell based vaccine.
So Denise, are you conceiving a combined antibody T cell vaccine? You've obviously got two programs. Are you thinking of combining them, or are these two different approaches, do you think?
At the moment, we are having two separate approaches. There are a limited number of antigens which are good targets of T cell and antibodies, but they're very limited. I think the most effective approach is probably to have a T cell based vaccine versus an antibody based vaccine, and then explore different ways of administering the vaccines in a human clinical trial.